Well, today we'll be discussing on the classification of bacteria and the classification of fungi. But before that, we'll see what actually is this classification, why classification is very much important to group organism, and what are the different classification schemes that are employed to group organism. So, as living organism is composed of a huge diversity of organism, it is desirable to classify or arrange them into groups based on their mutual similarities. Such an arrangement of organisms into groups is known as taxonomy. So we can also define this taxonomy as the science of biological classification, and it consists of three main parts. The first one is the classification, the second is nomenclature, and the third one is the identification. So we'll see what are all these three. Classification is the arrangement of an organism into groups based on their mutual similarity and evolutionary relatedness. Nomenclature is concerned with the assignment of names to the taxonomic groups in agreement with published rules. In short, we can say just naming of an organism which is going to be universally accepted. While identification is the process of determining that a particular isolate belongs to a recognized taxon. The dynamics of microbial evolution are so different with the dynamics of the evolutions of plants and animals. Hence, there is no theoretical basis for the assumption that microbial evolution has led to phenotypic discontinuities that would justify the recognition of a species. So for plants and animals that reproduce sexually, a species may be defined in genetic and evolutionary terms. But in contrast, a bacterial species is a group of strain that shows a high degree of overall phenotypic similarity and that differs from the related strain groups with respect to many independent characteristics. So next, we'll see the different classification system. So the first classification system to gain popularity is the Five Kingdoms system that was first suggested by R.H. Whitaker in the year 1960. This Five Kingdoms system contains Kingdom Animalia, Kingdom Plantae, Kingdom Monera, Kingdom Fungi, and the last one is the Kingdom Protista. The Five Kingdoms system is not accepted as there is a major problem in lack of distinction between Archaebacteria and the Eubacteria, and also the boundaries between the Kingdom Protista, Plantae and Fungi are ill-defined. Hence, the Sixth Kingdom classification is suggested. In this, it divides this Kingdom Monera into two groups, the Eubacteria and the Archaebacteria, and the rest remains the same. Later on, Cavalier Smith proposed the Eighth Kingdom system. He divides the two empires into 8 kingdoms. First empire bacteria contains Eubacteria and the Archaebacteria, and the second empire Eukaryota contains the sixth kingdom of eukaryotic organism. So we'll see the different classification schemes now. The first classification scheme is the phenetic classification. In this system, group organisms together based on their mutual similarity of their phenotypic characteristics. So the base phenetic classification is one constructed by comparing as many attributes as possible. In short, as many characters as possible. Next, we'll see the numerical taxonomy. Numerical taxonomy is a method used in taxonomy to determine and numerically express the degree of similarity of every strains of prokaryotes, and the information about the properties of the organism is converted into a form that is suitable for numerical analysis, and further this is compared by means of a computer. Further, the resulting classification is based on the general similarity as judged by the comparison of many characteristics. So under this, we can also calculate the percentage of similarity. This can be calculated as percentage similarity is equal to the number of characters that are similar by the number of characters that are similar plus the number of characters that are different. So next, we'll see the phylogenetic classification. In the phylogenetic system of classification, it is based on their evolutionary relationship rather than general resemblance. This system is difficult to classify bacteria and other organisms because of the lack of good fossil records. So, the direct comparison of genetic material and the gene products such as RNA and proteins overcome many of these problems. So now we'll see what are the characteristics used in taxonomy. So for classifying and identifying microorganism, the characteristics of the organism is studied. Characteristics are classified into two groups. The first one is the classical characteristics and the second one is the molecular characteristics. So now we'll come to the classical characteristics. Classical characteristics involves morphological, chemical, cultural, metabolic, antigenic, genetic, and finally the ecological characteristics. In the morphological characteristics, morphological features such as cell shape, cell size, cell arrangement, staining behavior, ultrastructural characteristics, and structure that are outer to the cell wall are employed. Morphological comparisons are very valuable because structural feature depends on the expression of many genes which are genetically stable and do not vary greatly with environmental changes. And in the chemical characteristics, it employs various cell constituent, cell wall composition, photosynthetic pigments, and so on. Chemical characters are useful in classification because they are directly related to the nature and activity of microbial enzymes. Now we'll come to the cultural characteristics. In this, it involves nutritional and the physical conditions that are required for the growth of the organisms. Now, in the metabolic characteristics, they are directly related to the nature and activity of microbial enzymes and transport proteins. They are also involved in energy production and energy utilization. Now we'll see the antigenic characteristics. Antigenic characters are concerned with specially large chemical components that we also call it as antigen of the cell. In genetic characteristics employs the hereditary material of the cell. Here, microbial genomes can be directly compared and taxonomic similarities can be estimated. Finally, in the ecological characteristics, these characters, that is the ecological characters, are valuable in classifying microorganisms because very closely related microbes differ considerably with the ecological characteristics. These characters involves the habitat and the distribution of organism. Now we'll come to the molecular characteristics. In this study or comparison of molecular characteristics such as proteins and nucleic acid yields information about the relatedness. Now first we'll see the comparison of proteins. Comparison of proteins determines the amino acid sequences of proteins with same function. The sequence of proteins with different function often change at different rates and so cannot be compared for taxonomic studies. If these sequences of proteins with the same function are similar, the organism possessing them are probably closely related. Next, we'll see the nucleic acid base composition. In this technique, that is the nucleic acid base composition, the DNA base composition is determined. And we all know that in double-stranded DNA, adenine pairs with thymine and guanine pairs with cytosine. Thus, the guanine plus cytosine by adenine plus thymine ratio or percentage of G plus C in the DNA reflects the base sequence. If two organisms they differ in the G plus C content, that is the guanine cytosine content by more than 10%, their genomes have quite different base sequences. Hence, G plus C data are taxonomically very valuable. Now, we'll come to the nucleic acid hybridization. Nucleic acid hybridization is used to compare genomes similarity between organisms. In this technique, the double-stranded DNAs of the organisms are mixed and it is heated to form the single-stranded DNA. Further, it is cooled down below melting temperature. At this temperature, the complementary base sequences reassociate to form the double-stranded DNA. If these DNA molecules are very different in sequences, then they will not form a stable, detectable hybrids. Next, we'll see the nucleic acid sequencing. RNA sequencing are nowadays more extensively used in microbial taxonomy. From among the RNAs, the rRNA are most ideal for studies of microbial evolution and relatedness because they are essential to critical organelle found in all microorganisms. Their functional role is same in all the ribosomes and their structure changes very slowly with time. As this ribosomal RNA contains variable and stable sequences, both closely related and very distantly related microorganisms can be compared. Now we'll see the bacterial classification. Earlier in the year 1928, David H. Berge published a classification of bacteria that could be used for identification of bacterial species for the first time. And these came to be known as Berge's Manual of Determinative Bacteriology. This classification system, given in the first edition of Berge's Manual of Determinative Bacteriology, is mostly phonetic, and the characteristics that are used to define sections are morphology, gram staining, oxygen relationship, motility, presence of endospore, mode of energy production, and so on. Under this, bacterial groups are divided into 4 volumes. So in the first volumes, it contains the gram-negative bacteria of general, medical or industrial importance. Second volume contains gram-positive bacteria other than actinomycetes. In the third volume, it contains the gram-negative bacteria with distinctive properties, cyanobacteria and the archaebacteria. In the fourth volume, it contains actinomycetes, that is the gram-positive filamentous bacteria. Further, bacterial taxonomy has enormously progressed after the first volume of Bergey's Manual of Determinative Bacteriology had published. And also, the number of named species has been doubled, and there are well over 170 newly described genera. And the sequencing of ribosomal RNA, DNA and proteins has made phylogenetic analysis of the bacteria feasible. As a result of this, the second edition of Bergey's Manual of Systematic Bacteriology is largely phylogenetic. In this second edition, the second edition is published in 5 volumes. It consists of more ecological information about the individual. It does not group all the clinically important bacteria together as the first edition had done. Instead, pathogenic species are placed phylogenetically and thus scattered throughout the 5 volumes. So now we'll see the 5 volumes. So in the first volume, it contains the archaea, cyanobacteria, green phototrophs and deeply branching genera. In the second volume, it contains the proteobacteria. Third volume contains the low G plus C gram-positive. In the fourth volume, it contains the high G plus C gram-positive. Finally, in the fifth volume, the planktomycetes, spirochetes, fibrobacter, bacteroids and fusobacteria are incorporated. Now, we'll come to the classification of fungi. Fungi are eukaryotic, spore-bearing organisms with absorptive nutrition. There is no chlorophyll and they reproduce asexually and sexually. Based on 18S rRNA studies, modern microbiologists classify fungi into zygomycota, the ascomycota, basidiomycota, then deuteromycota, which is also known as fungi imperfecti, and finally the chytridiomycota. So now we'll see the division zygomycota. Zygomycetes fungi belong to the division Zygomycota. They live on decaying plant and animal matter in the soil, and some are parasites of plants, insects, animals and human. One of the example is Rhizopus stolonifer, which is commonly known as bread mold, is a member of this division. They reproduce asexually, but at the time of scarcity of food or at the time of unfavorable environmental condition, they reproduce sexually. Zygomycetes, they also contribute to the human welfare. One of it is Rhizopus is used in the production of tempeh. So tempeh is nothing but it's an Indonesian food products that is produced from the boiled skinless soybeans. Another one example is the mucor. This mucor is also a zygomycetes which is used in the preparation of cheese that is known as sufu. Not only this, zygomycetes are also used in the commercial preparation of anesthetics, birth control pills, industrial alcohol, meat tenderizers and so on. So next we'll come to the division Ascomycota. In this, it contains the fungi Ascomycetes. This Ascomycetes is also known as sac fungi. Ascomycetes are named for their characteristic reproductive structure that is known as ascus. They reproduce by both asexual and sexual reproduction. But asexual reproduction is common and takes place by the way of conidiospores. Sexual reproduction involves the formation of ascus containing two or more haploid ascospores. So fungi under this group includes the red, brown and blue-green mold and are responsible for various food spoilage. Many ascomycetes also cause disease known as ergot in rice and other grasses. So that was about the ascomycetes. Now we'll come to the division Basidiomycota. Basidiomycetes is commonly known as club fungi. Their hypha consist of both monokaryotic, here monokaryotic means it contains only one nuclei, and dikaryotic. Dikaryotic is it contains two nuclei. So some of this Basidiomycetes includes smut, jelly fungi, rust, cell fungi, stinkhorns, puffballs, toadstools, mushrooms, etc. And majority of the Basidiomycetes, they reproduce sexually, producing a club-shaped reproductive structure that we called it as basidium. And for asexual reproduction, mycelium becomes fragmented to form separate individual. And some of these Basidiomycetes are edible and some are deadly poisonous. Few of them are pathogens of plants and some are very important decomposers. So now we'll see the division Deuteromycota. This division Deuteromycota is commonly known as fungi imperfecti. They are mostly terrestrial and are either saprophyte or parasite of plants. So these Deuteromycetes cause human diseases such as athlete's foot, ringworm and histoplasmosis. Deuteromycetes are also industrially important in the production of antibiotics. Antibiotics includes penicillin and grisofluin. They are also used in the production of cheese such as gorgonzola, camembert and roquefort. They are also used in the production of citric acid, glucuronic acid and the gallic acid. Next, we'll see the division Chytridomycota. These Chytridomycetes are the members of this Chytridomycota and they are commonly known as chytrids. Members of this group are terrestrial and aquatic and they reproduce asexually by forming motile zoospores with single posterior whipless flagella. For sexual reproduction, zygote formation takes place and becomes resting spores or sporangium. So some of the members of this group grow saprophytically and they are parasite of algae. So that was all about the classification of bacteria and the classification of fungi. And I'll conclude today's discussion by stating that grouping of similar organism and organizing this group in a non-overlapping hierarchical arrangement is quite necessary to make the diversity of organism sensible. And the basic taxonomic group, which is known as species and which is defined in terms of their sexual reproduction or general similarity. Although Burgess' Manual of Systematic Bacteriology provides the accepted system of bacterial classification, bacterial taxonomy is rapidly changing due to the acquisition of new data by the use of molecular techniques such as ribosomal RNA structure and chromosome sequencing. As a result, new phylogenetic classification emerged.